Okay, I guess we started, right? Let's say, yeah, people trickle in. I still missing John, but we'll play by ears. Let's see what happened. I guess the worst case scenario, Angus and I can cover his part, and I already listened to his webinar, so I can just fake, every core analyst, I guess. Anyway, I think we should get started. I mean, it's sorry, this is not good. Go away. All right, I guess we should get started. First of all, thank you everyone. This is a Friday night in the US and also Saturday morning in China or in Asia in general. If you are here, that means that you're really dedicated to learn what's really going on and ASCO this year. I hope we will not disappoint you. We are today, if you had attended our ASCO preview last Tuesday, which by the way, we attracted around 5,000 live audience. Today, we hope many of them who had attended that one are coming back to listen to whether we were right or we were wrong. And that's going to be fun exercise because we're definitely gonna, show you our forecast before the ASCO, and we can revisit what's really going on after ASCO. So today, unfortunately, actually, Dr. Chandran Park, due to family issue, he had to drop out. And also, we are still waiting for John Miller. He may join anytime from now. But with no further ado, let's just quickly reintroduce the panelists today. Actually, before that, let's just give a little bit introduction of the three co-organizers of this event. As you can see here from our the landing page, this event really has been driven by instance trust community, very young community. We are on the way to become nonprofit, really focus on ethnic Asians in the biotech industry globally, particularly in the, I would say, business investment, the business and also commercial marketing aspect. Of course, many of the members were R&D to start with, so that's definitely our DNA as well. The second organization is the BioSpark, which was founded many years ago in Boston, but also have members around the world. The third one, obviously, is probably the oldest and the most established ethnic Chinese pharmaceutical professional organization called Sino America Pharmaceutical Professional Association, SAPA, particularly a Greater Philly chapter. This is our third collaborator. And we also want to thank our media collaborators, 5 of them. They are broadcasting this event live. Probably they are reaching thousands of audience right now. And now move to the speakers. First of all, Dr. Peng Li. She's a Chief Scientific Officer of Palian Pharma, which is upcoming next generation immuno-oncology biotech company with their lead assets already in clinical study. And their scientific founder, Dr. Caroline Bortozzi, won Nobel Prize exactly the same technology, which is a click chemistry, which she used to lay the foundation of Palian Pharma. That's for Dr. Peng Li. The next speaker is Angus Liu. He's a senior writer with the Fierce Pharma. He has been writing about the biotech and biopharma industry for many years, and I think he has a pretty decent size of followers right now in the pharma industry, and he has a lot of really insightful articles interviewing CEOs, FDA commissioners, and all those key decision makers in our industry. And by the way, he is also a volunteer of Instance We Trust community. Then I will save John Miller later once he dive in. A little bit about myself. I'm a founder of Instance We Trust community and also advisor to BioSpark. That's why I kind of sit in both organizations. For myself, for my daytime job, I'm also the founder of Instance We Trust at a BioAdvisory, which is a cross-border business development advisory firm. That's my primary job. That's where I get my paycheck. With that being said, with no further ado, let's move on into our webinar. Whenever John Miller get online, please, whoever in the background, give me your notes so we can, bring him into the discussion, because we really need his insight from the investment bank point of view. This is how we're going to do this today. This is actually this is the exactly same slides we published on May 30th when we did the preview. That's what that's our record. That's the prediction before ASCO started. And just give you the new audience members a little bit explanation, how does it work. So pretty much before ASCO started, we selected some of a high profile abstract based on our individual, best knowledge. And then we made a prediction, how likely they're going to be successful in terms of the context. Obviously, if they are positive, give them the really dark green color. If they are kind of a slightly positive, give them like teal color, positive, neutral. If they are kind of we think probably going to be just a very mixed, not necessarily going to move the needle, we can have the neutral. If they are pretty bad, leave negative, neutral, negative, which is pink color. And finally, if they are really disaster, we give them negative. But obviously, nobody know which one going to be really super negative. So that's what we had. That's what we threw out there. We are willing to to be corrected. So now ASCO already in the past. Let's go to revisit this. Say how right or wrong we were. This is pretty much what we're going to do this. And also I put the headshots of each one of us who actually made a prediction in that category over there. If you want to see the detail who specifically made a prediction per abstract, you have to watch our replay of the preview. That's going to be a fun exercise. So without further ado, as you can see here, the title today, we really talking about the winners, losers, and the black horses. Obviously, the winner and loser is going to come from the pre-selected abstract we did in our preview webinar. And we also identify a few pleasant surprise we're going to share with you later on as well. So let's go to those three buckets one by one. I think we should we can get started with the let's say the first bucket. That's actually how we're going to do this. As you can see here today, the new game is, we already know the color. We assign them, pretty much the results we forecast or predict they're going to be. Now the results are already there. Now let's look at the results, look at what they presented in ASCO, then decide how right we are in terms of prediction of their outcome. So if you are spot on, we give the hit, which is, this lovely red color we're going to put on top of the abstract we just said, predicted. But of course, given a lot of audience are new to this webinar, I would suggest whoever is going to take a lead of this abstract, please describe the results and what the chatter, what's the insights you may share with the audience. And then, we can assign, this kind of a trophy score on each of the abstract. So unfortunately, John Miller's still not here. I guess, Angus and I gonna, try to replace him. We always do this. Unfortunately, it's a Friday night. A lot of colleagues probably they forgot because they have a lovely dinner or drink. It's a long week. I mean, I don't blame them. It's like, ASCO plus bio. A lot of us, totally exhausted, but we are still kicking. So with no further ado, so we'll start with the first one. Abstract 4010. So Angus, do you want to take this one? You want me to take this one?
Leon, feel free to take this one. I might want to add a few things afterward.
Okay.
Go ahead.
Okay. So this one really is actually John select this one, so his top pick before the preview, and the turnout is more or less lived up to the expectation. So data hold up pretty well. That has a ratio, everything exact as an abstract show. And their own to just give you a little bit of background. This is from MORPHUS-Liver, which is I would say it's kind of a umbrella trial, if my terminology is correct. It's pretty much based on the different biomarker, the MORPHUS-Liver, each arm would receive a different combination of treatments. Most of them are have like PD-L1, which is atezolizumab. And for this particular one, what they got is they have a triple combo, which is their PD-L1, which is tislelizumab, and their TIGIT, which is tiragolumab, and their VEGF-A, which is bevacizumab. The triple combo, and in comparison with the standard of care right now in liver cancer, which, as you know, Roche also get approved a few years back. And from this one, the hazard ratio is really good. It's really, I believe it's like a lower than 0.5%. Sorry, 0.5, not so that's not 0.5%, 0.5. So it's pretty good. And the only criticism a lot of people had is that the experimental arm actually underperformed compared to the Roche's own pivotal trial. I believe it's in Brave. And so, however, the pushback from there's pretty much there's two camps, right? Some of them believe this is good data. They believe that because despite the control arm in this particular trial underperformed versus the other pivotal trial, they said this is the internally controlled. Probably the patient population, all those kind of treatment history is different. As long as they were internally controlled, the patient baseline characteristics between the experimental arm and the control arm had no huge difference, they should be okay. So that's one argument. However, on the other camp, they also pointed out that, well, the cityscape had a similar spectacular hazard ratio. I believe that's a 0.29 measure if you use a PFS, but they also have spectacular failure. As you may recall, the skyscraper, they're really, really spectacular failure. So people worry that, the negative camp is like, this may be just like, yesterday once more. After some discussion with quite a few KOLs and also the analysts and also listen to the abstract, I think overall people still think this is could have some leg in the signal. This is pretty much, I think, in the end, after people saw the data, they still give the pretty positive. I mean, one strong indication is that if you look at all the TIGIT, companies, especially like Acros and many others, their stock just traded up significantly, and they stay there more or less even after the presentation of the data from MORPHUS-Liver. So I guess that's a very good indication that the investor believe there's probably have some leg this time. I'll stop here. So, Angus, do you have anything to add?
Yeah, I just wanted to add that, yes, we know before the official presentation, we know that this is good data, but we also know that the relatively underperformance of the control arm. But during a panel discussion I was moderating in Bio about checkpoint inhibitors, Ian Waters, he's currently the CEO of Portage Therapeutics, but he was previously with BMS, working on Opdivo and Yervo, this stuff. He mentioned that he was kind of pleasantly surprised because he noticed some very positive prognostic factors in the control arm, which the experimental arm, the TIGIT arm, didn't have, meaning even though the control arm had some positive signs for the patients, they still performed that way, whereas the experimental arm didn't have those favorite factors, but still showed that massive improvement over control arm. So that might mean we actually saw a real signal, a true signal there, positive signal there for the TIGIT arm. But on the flip side, he also noticed that just because liver cancer is very complicated, even in a very well randomized study, you still see some of the imbalances in patient characteristics, and sometimes we don't fully understand them. And he was actually still cautious about translating this finding into like repeating it, seeing the same kind of finding in a phase 3 trial. So I think for me, I read a lot about e-market products, so I'm more of a show me the phase 3 data kind of guy instead of making too much speculation based on phase 2 studies. So I think we'll just have to see, and maybe just not trying to prematurely translating this into potentially other cancer types. this is just liver cancer for now. That's it for me. Leon, you're muted.
So Angus, sorry, I muted myself. I agree with you that the biology between liver cancer and small cell lung or even non-small cell lung could be very different. For one, as an immunologist by training, I know like liver cancer have a lot of, if you may, myeloid cell, which is called a Kupffer cell in liver, which is the, immunosuppressive. That just tell you the biology between, let's just say, non-small cell lung cancer and the liver is quite different. Let's assume if the signal is real, probably does also tell you TIGIT is not as broadly active as people thought. That probably really depends on specific tumor microenvironment, which is tied to a specific indication, right? So if that's the case, the good news could be that if this is a real signal, that means that probably the TIGIT molecules really work well for kind of myeloid-rich cancers. But the bad news is that probably the commercial potential is not as big as people thought. Let's say, for me, someone has been doing early stage clinical assay evaluation. I think this, again, I agree with you, from phase 2 phase 3 is a huge gap. But this, I think the data is good enough to justify to open pivotal. Obviously, data has no way, as some people probably don't understand, though this is going to be sufficient for submission. There's no way. This is definitely not sufficient for any submission. Just forget about this. I don't think Roche can do this. No one, like their right mind can do this. So with that being said, do you think we more or less hit on this one before we move on?
I think it's still positive. Yeah.
Okay. Do you want to take the next one? You want me to take the next one?
Go ahead. I will probably just take the ribociclib one.
Okay. I'm glad I watched like every course ASCO review, so I pretty much more or less know what John Miller would have said. For this one, it's very interesting. So actually, it's voting to right now, I would say one of the hottest topic in this year's of ASCO. Not just like, Kelun and Merck presented their, I would say phase 1b data of their top ADC. Obviously, Gilead presented their data, and also AstraZeneca Daiichi presented their data. So this is going to be a very competitive space. Right now, I can tell you the data, it's hard to compare because their patients are different. Just for this, we can go to the nuances that are different. But one thing pretty much everyone agree, Gilead Trodelvy probably is not the best drug. But they obviously have a head start. They get approval quite a bit ahead of everyone else. But the truth is that they get approved in triple-negative breast cancer. So in like a non-small cell lung cancer, that's a fair game. So more or less everyone, probably under most 2 years ahead of each other, like apart from each other. Which, based on, again, this I just quoted John directly. He said, if there's any company who can catch up after 2 years behind, probably that would be Merck. Merck's clinical operation, fortunately for them, unfortunately for their competitors, is very, very, I would say, dominant. Just look at the story between Keytruda and nivolumab, what we are talking about. So that's something they are having a pretty good track record. And their data, you put a totality together, the conclusion, kind of consensus is like, they're comparable between the Merck Kelun and I believe this is Dota DXD, which is top 2 ADC from Daiichi and AstraZeneca. So this is going to be a very interesting showdown, if you may, between this probably the 2 biggest oncology players. Right? Each one of them have a decent weapon, but none of them have a clear dominance over the other. Of course, there's a head of like a clear leader, but a little bit weaker. Trodelvy in front of them, they're going to be very interesting dynamics. But so in the end, I think this market will not be kind of winner take it all, at least in the foreseeable future. Going to be a lot of nuances. The execution, the selection of an indication, how fast they, how do they gonna market their the drug once they get a first approval? What's their, kind of a label expansion experiment going to be? And on top of that, there's one big elephant in the room, which is the combination with the PD-1, right? Obviously, that's a big, kind of a indicator or also a factor. In that sense, obviously, Merck has a little bit advantage. They have a probably most established PD-1 franchise. and they have a lot of, I would say, stickiness from the physician community. On the other hand, AZ has been going strong, really, with a lot of momentum. The ADC franchise is absolutely, dominant right now. So, going to be very interesting. But of course, like I mentioned, Gilead is ahead of a little bit ahead of both of them with a little bit kind of interesting molecule. So, going to be very interesting for us to say. I think there's going to be opportunity, going to be a multiplayer market right now for the TROP2, both in non-small cell lung as well as in triple negative breast cancer. So that's what I would say. So I think our assignment is like a kind of like a positive neutral is fair, because the data is good, but the data is not a kind of a give a Merck and Colon sort of advantage of all the other two players. That's what I thought. What do you think, Uncle?
Yeah, well said, Leon. I think I agree with most of what you said. I just wanted to point out that this is a study that enrolled some of the EGFR mutant non-small cell lung cancer patients. I think this would be a very good place to start for this TROP2 ADC, in that we just also at ASCO Merck detailed this clinical trial, Keynote 789, I think, where Keytruda plus chemo failed in EGFR resistant non-small cell lung cancer. So this could be a TROP2. I think Merck has plans to start a global phase 3 study in this post-EGFR TKI setting. Just in this EGFR resistant, I mean, this could also be a big market if it eventually succeeds here. And it appears that in EGFR resistant population, these agents appear to work better. I think just generally these TROP2 agents work generally better in EGFR mutant non-small cell lung cancer. So I think that could be a good place for this class of non-small cell lung cancer. That's it from me.
Okay, sounds like you and I agree. We actually made a pretty good prediction. we believe there's a good data, but it's not like a home run. Okay, another how to say, pat on our own shoulder. You want to take the next one? That's something you wrote a lot about. I know you are the first one actually published in the morning.
This is obviously a lot of the focus of Novartis and behind this. Just a reminder. This is Kisqali, ribociclib's NATALEE study compared with Eli Lilly's Verzenio, abemaciclib, Monarch E study. We're comparing these two different studies here. Both studies, the one presented as ASCO is Kisqali's NATALEE study, but both studies are stage 2 to 3 early stage HR positive, HER2 negative breast cancer. But the difference being Kisqali's study, the NATALEE study, also included patients who have no lymph node involvement, whereas the Monarch E trial included only node positive patients. So about the data, we see the NATALEE after median 34-month follow-up, there was a 25% reduction in invasive disease-free survival, which was numerically below about 30% reduction Verzenio showed after a shorter 27-month follow-up. And as we know, because Verzenio has longer follow-up time, we know later on Verzenio's IDFS benefits widens as you follow patients longer. So this kind of, even though NATALEE has a longer follow-up, it's still numerically below Verzenio is kind of a little bit concerning to me, but they're not that different. Let's just put it out there. It's not that different between the two. And again, it's not an apple-to-apple comparison in that NATALEE included a wider patient population. And if we look specifically at node-positive patients, Kisqali had a 23% IDFS improvements. Again, this was numerically below the Verzenio study. But there is a very positive sign from the NATALEE study, I believe, which is a death risk reduction of 24%. That is a very positive sign, given that Monarch E hasn't shown a very clear OS benefit. There is still not as clear at least as we see here in Natalie. But again, Novartis is very keen to point out that it only has, at this point, it only has about 20% of patients in Kisqali have finished treatment. So it's very interesting to hear Jeff Flacco said Novartis say this, because on the one hand, he wants to say this is a little bit immature for this data comparison with Verzenio, but on the other hand, he said this is mature to ask FDA for an approval. So immature for data comparison with Verzenio, but mature enough to ask to seek approval. Now, there is another one key difference between the two studies is that Kisqali, the Kisqali Natalie trial, the Novartis trial used a lower dose of Kisqali to try to avoid some of the side effects, but instead used a longer treatment duration of 3 years, whereas latest trial used a 2-year treatment duration. I think the investigator and Novartis, they were behind this rationale for longer treatment, in that they argued that longer treatment send more cancer cells into a senescent state where they stop growing, stop dividing, and you could potentially see a better outcome over the long term just because you have sent those cells into the senescent states. So again, they wanted to see a longer term data to really tell a difference with the elongated data. And then, of course, on the safety, they lowered the Kisqali dose to kind of tease out a safety benefit here. We did see some safety benefits for this lower dose, lower neutropenia and slightly lower, I think, what was the other side effects? Well, generally, some of the key important side effects were lower in this new Natalie trial versus some of the historical studies for Kisqali in the metastatic setting. So I think they did achieve this in terms of better safety profile within Kisqali itself. But again, I think at this point, it's very hard to tell, just because of the similarity between the two data set, who will eventually succeed, which one of the agents. So I might want to maintain my neutral, but slightly positive than I was before the preview. So just because it has that OS benefit, and because I think I am slightly going to buy Novartis' argument that this is they need some longer term to really show that benefits of a longer treatment duration. But that said, I think this will again come to commercial execution, just because you have so similar data sets. You're not going to tell all the nuances of the difference between the trials. You can't persuade talk these to almost all the other physicians beyond those large academic centers. You just have to take them by face value, and right now your face values are very similar. So I don't think this is very going to shake up the Eli Lilly's market very much, but there might be a chance for Novartis to make to take more market share if they have longer term data. But right now, I think it's a close call. It might come down to commercial execution. That's it.
Yeah. So I guess I was about to put a mix, but I guess it sounds like in totality of from what you said, is they're pretty much, people cannot say they are better than Eli Lilly, right?
I'm just slightly more positive on that spectrum of neutral. I'm slightly more positive than I was.
I guess we hit on this one, but one thing really caught my eye. A lot of people talk about is they have this QT elevation, right?
Right,
something like that.
interval.
So that's actually
this is a
really
known
worry
side
a lot of
effect
physicians.
ribociclib.
Yeah, this is like those are low risk patients. You give them QT elevation. That's not something they like.
Yeah, you have to remember Kisqali also has a GI toxicity, which is very symptomatic. You need QT intervals, so you can manage, you can monitor it, and I've heard physicians said this is not too much of a burden, they can manage it. But GI side effects, that's very obvious to a patient. That could be a problem for Verzenio, and it's So they have their own side effect problems. It's hard to tell whether who's gonna take what kind of patients, who's gonna take what share. So might be just staying the same at the moment. No major disruptions.
Talking about GI tox, actually, I heard a lot of doctor told me GI tox actually really kind of a link to poor compliance of the treatment, because if you are not like a high risk patient, that pretty much means if you go out, let's say right now summertime, go out for fun, you have to make sure you always have some toilet 5 minutes away from you. That's just very, very annoying. Just to give people a bit idea what that means. When we say GI tox, that's pretty much means that you need to go whenever and you don't have time. Okay, so let's move on. Next one actually, I guess I already can tell you I'm gonna put a mix because we thought it's gonna be good data. Turned out it's not that good. I learned a little bit more. As you know, Angus already mentioned, the CDK6 for Novartis is kind of meh. But this one, Pfizer obviously also have their own CDK inhibitor. They want to pretty much, they want to expand their franchise, which is they are losing, to be honest. They're not that great. So they have this slightly different kind of a flavor of a CDK, which is CDK4. Initially, I thought the data looks good, but what happens, their tox is pretty bad. Actually, that's number one. Number two, I learned something which I didn't know before. I don't know whether Angus know. So if you, let's say a patient progressed on even like CDK6 inhibitor, you let them rest for half a year, you retreat them again, about like 20% of patients gonna respond. The same drug. So when I heard about that, okay. Yeah, I was like, okay. So yes, those patients progress down traditional standard of care CDK inhibitor.
Yeah, I've heard physicians talk about sequencing CDK4/6 inhibitors just among those. They might use Novartis, Kisqali. From what I've heard, they might use Kisqali first because it has shown an OS benefit in the metastatic setting. And just because it is more effective, physicians think it has a higher toxicity. And then after that, the second line, they might start using another CDK4/6 inhibitor. Yes, I mean, retreatment with CDK4 inhibitor is a thing, definitely.
Yeah, so if that's the case, I kind of worry about their value of this guy. Yes, they have like 10% higher. By the way, this is the phase 1, right? Phase 1, you always have the best data, as we know, and your data just drop, unfortunately. Historically speaking, statistically speaking, over time. So let's assume they're gonna be in the same neighborhood of 20% when they get into pivotal. So I'm not sure a physician can really buy this. The physician is not a kind of a PhD scientist, right? They want to use a new drug. Actually, they hate new drug. If old drug do the same job, they prefer to use old drug, right? So with that being said, actually I think we were probably a bit too positive with this one. This one just neutral. There's something Pfizer can talk about. They say what's gonna happen, but so far the current data doesn't look they really would be competitive. Really gonna kind of do the job as a Pfizer expect this molecule gonna do. But anyway, you never know. Let's say what they're gonna do afterwards. This is the first in class, right? First in class, there's probably always some feature. They're gonna find some niche, but still, it's not as positive as we thought. After I know there's a kind of a sequential treatment of the patient with a different kind of a company's CDK inhibitor, actually they're gonna respond. So that's something I also learned. That's good for me. Anyway, that's for me. Next one, I guess I can also take. This one, I think next one actually we hit. It's very interesting. So this is actually a really good segue to the BCMA in general. This is kind of a little bit of teaser of what we're going to talk about Carvykti. This is just tell you BCMA space is so competitive. It's unbelievably competitive. You really have to, the benchmark is so high, let's put it this way. If you put like accessibility and the logistics aside, Carvykti is such a high bar. Look and we're gonna talk about data later on. They have 99.4% in second line ORR. It's pretty much almost like 100%. There's one, when you're talking about 99.4%, that means like only one patient failed for some reason. There could be anything, and their duration of response is pretty long. Now go back to this one. This one is not that good. First of all, your ORR is a little bit lower than Carvykti, number one. Number two, they have a DOT on the first level, dose level. That's already pretty bad. If you look at data they presented over there, so if you look at totality, yes, Carvykti, the problem is Carvykti is pretty safe, also effective. The only challenge for Carvykti is right now, the manufacturing is the bottleneck, right? That's really, if you compare this combo to the other one, in terms of a clinical profile, it's inferior to the other two. To Carvykti in the same indication. And actually their toxicity, like CRS and also neurotox, especially infection, it's pretty bad. It's not something you can easily handle. So then once you reduce the dose, it probably is not that good. So I think actually I would say this guy would definitely hit it. It's not as good as people think. We'll see how they're gonna do this. I understand why J&J is doing this. J&J obviously right now is dominant player in multiple myeloma. They want to build their franchise, if you made their moat, right? Have multiple layer of protection, build their kind of pretty strong presence in multiple myeloma. This is part of their strategy. The truth is, even this combo probably is still better than many other pharma's, other players' drug. Just take a GSK's BCMA ADC. This is definitely better than GSK's BCMA ADC. So that's also tell you the bar in this space is pretty high. I understand why they're doing this because it's kind of two different targets, right? They kind of probably address a lot of potential problem. But so far, the initial debut of this combo is not that encouraging, I would say. They may tweak it, but so far in the current shape, I think they still. I don't think they're going to take the current shape into phase 2 pivotal or something like that. That's my opinion. What do you think, Angus?
Yeah, I think that just having a Carvykti-like data set isn't enough at this point. You have to have something else to offer beyond Carvykti to really get any meaningful market share. I mean, I agree with you. This is definitely something that not as, at least at this point, not as promising. That's what I would have hoped. Yeah.
So the last one, actually, we're gonna upgrade. That is really, it's John's like a specialty. I don't know this drug very well. I don't know the target very well. I apologize. We're gonna move on. So this one we're gonna be ungraded. But the good news for everyone, number one, just give me home housekeeping. We do have a Q&A button. If you do have a question, please feel free. Type your question in the Q&A. Should be in the bottom of your screen. Type in there. Some of our, I would say our volunteers gonna take a look at them. Also, probably some of us, one of our speakers, 3 of us, gonna reply to your questions by typing. If they're really good one, I think we can address live. But that's number one. Number two, right now you probably say, yeah, what you guys are doing? Well, there's no slides. Good news, we now have slides. From now, the second part, we do have slides. The second part actually is about the rise of Asian biotech. This is a 6th abstract that we selected. We're gonna go one by one, and we did include the key data point from each of those abstract, and the same can apply to the third bucket, which is practice changing abstract. So Angus and I gonna have slides to show the key data show as well. So now you can have a more kind of a visual information from us. With that being said, actually the first one actually come from Carvykti. Already mentioned that BCMA is super duper competitive. How competitive it is, let's look at the no doubt clearly best in class BCMA drug in this space. Very likely could be the best in class multiple myeloma drug, because as you know, they are in front line clinical trial as well. So just look at the second line. As you know, the data already know the key data. Now you look at the nuances, actually slightly better than people expected, at least in my opinion. First of all, hazard ratio already know the disease progression reduced by 74%, so hazard ratio 0.26. That's number one. Number two is not only the hazard ratio is good. Also, if you look at the ORR, obviously there's a 2 way to measure their ORR. Number one is like an intent to treat population. As you know, once you expand to so many patients, such a large scale thing happen. So as you know, like autologous CAR T does have the problem of manufacturing and the logistics. So sometimes the patient could not get the drug. So that's why, you know, if you look at intent to treat the population, the OR is 84%. However, if you look at the patient who were treated, it's 99.4%. So pretty much all the patients treated, probably only one did not respond for some reason. You know, that's just tell you the Carvykti, their efficacy hold up. And that's number one. Number two, if you look at the toxicity of Carvykti in the CARTITUDE-4 versus CARTITUDE-1, they actually improved. I mean, that's I guess because you know, after they have been using this drug for a long time, they probably improved or further improved their CMC, also further improve how they manage the toxicity, how they give the drug. So they just become more experienced. So it turned out their toxicity profile in CARTITUDE-4 is better in CARTITUDE-1, which is good news. Now also, you know, definitely they're gonna help the penetration of this drug in, you know, in broader patient populations, right? Especially for some communities where they do not have those fancy ICU or neurologists on board, that can definitely help them. So that's a very good news. But this also tell you, you know, a lot of you know, experimental drug based on the premise that Carvykti or CAR T would have a you know, kind of unmanageable toxicity in outpatient setting. Probably have to think again. I think they're getting closer and closer that this drug can be given in outpatient setting. If that's the case, the only thing, you know, kind of prevent the Carvykti really penetrate all the reach all the eligible patient going to be their manufacturing and their logistics, which still there. I don't think the current shape and form of the drug can solve the problem. So there's still some room. But I guess that kind of you know, wrong and getting smaller and smaller. That's what I can say. So, you know, for me, this is a clear hit. You know, as we expected, it's a really, really big hit. And also this totally changed the paradigm of the multiple myeloma space. And right now, if you are still doing BCMA or even CAR T for multiple myeloma, you really have to ask yourself a hard question. What differentiated factor do you have compared to Carvykti, which can only get better? If you just look at their trial from CARTITUDE-1 until CARTITUDE-4, they're getting better and better. Because as you know, the cell therapy, unfortunately, there's a lot of know-how in terms of their manufacturing as well as their, let's say, utilization in the clinical practice. So that, you know, fortunately for Legend and the J&J, they have a head start. They are gaining a lot of in the field experiences, which the other competitors in later, you know, they are still in pipeline, they have treated fewer patients, don't have it. They just have to admit that. So for me, I would give a clear hit. So what do you think, Angus, before I move on? Oh, sorry, actually, this is Peng Li, Dr. Peng. What do you think? Do you agree or disagree before I move on?
Oh, I echo what you said. Definitely hit.
Okay. I just wanted to add one thing, Leon, if it's okay.
Please go ahead.
Yeah, the Carvykti data itself, efficacy, safety data themselves are definitely a positive, but there are some side notes from all this that J&J and Legend sure need to work on, which is on the manufacturing side of things, because we're seeing a turnaround time, the CAR T turnaround time that's very long for Carvykti, that hearing frustration from physicians that this is taking too long, especially compared to their BMS competitor. I think that they're seeing a turnaround time for Carvykti over 2 months, whereas a big month, the BMS data was about a month, slightly more than a month. So it's the turnaround time. Of course, if you look at it as treated patients, this is very spectacular data, but there are patients who, because of your slower turnaround time, progress. So that is something that J&J and Legend definitely need to work on. But still, on the flip side, I'd also like to say that as you move to the earlier treatment line, this turnaround time might not matter that much, because if in later line patients, they relapse very fast, you need to turn around this drug fast as well. But in an earlier treatment setting, this might not matter that much. That said, it will be still good to have that turnaround time and also those manufacturing failure rate down and improved. That's all I want to say.
Yeah, I guess that they are trying to fix the problem. They already kind of have a partnership with the Novartis to kind of get their capacity, but that would not be in like in practice or like help them. Yeah, several years, right? But they are working on this. So I guess right now they still have to suffer from their bottleneck of manufacturing. That's what we know. But the data looks pretty good. That's
of course, yeah.
All right, I'm gonna move on to next one. So this one's a hit. So this one actually is from Gracell. It's a China cell therapy company. They're based in China. They also have operation in the US. This is the NASDAQ listed cell therapy company. I mean, we heard about this program and we saw the news and everything looks pretty good. And now we saw the data. And so if you just look at the data, I would say if you compare this to CARTITUDE-1, it's largely comparable if you look at the clinical data. As you can see here, the Carvykti is almost a 35-month as of today. I actually presented at ASCO this year. So it's very easy to compare. As you can see, this is the CD19 BCMA dual CAR. They have a similar, I would say, PFS or duration. PFS in this case, very similar. So that's one. Also, if you look at the OR, also very, very comparable. There's just not much you can really improve, right? There's pretty much between 98% to 100%. It really doesn't matter if you are 98%, 100%. It's really not a big deal. This all look good, but we just have to say there's one thing, that's not from me, but from the analysts that they repeatedly mentioned this. This is the China-only trial. But you can argue Carvykti, when they were doing experimental pipeline drug, when they have 100% ORR initially, done by Nanjing Legend right at that time, there was also China-only data. So there could be like yesterday once more, right? The same thing. In addition to that, one thing I want to mention that this is the CD19 BCMA CAR actually can be used for not only for multiple myeloma, can also be for B-cell non-Hodgkin lymphoma. So they actually also have data presented on ASCO, if I'm not mistaken, it's 100% OR. That looks kind of a supporting evidence to show that this really has something. In the end, I really want to emphasize I would say compared to Carvykti, the huge, the biggest. Oh, John, you are back. Hey, John. Hello. Can you hear me?
I can hear you.
Yeah. So let me finish this, and we just fake your role. We just part through. We actually should go back to talk about the V1. I just apologize to the audience because I really don't know anything. I don't know anything about it. I'm gonna go back to let you talk about that. Let me finish this first. So you say the whole party's here. Even later is better than never, right? Always appreciate people, especially Friday night. Anyway, let's finish this one. Please correct me, by the way. So you actually covered this. I'll try to repeat what you said. I largely learned from your ASCO review. So this guy, this one, I remember you said, all good, comparable, but we have to just mention that this is China-only data. Doesn't mean it's bad because the Carvykti, in the early days, they also have 100% OR from China-only data. They get to reproduce. So, but just something you put a little bit additional scrutiny, look at the individual patient characteristics, make sure they are kind of comparable to the patient population to CARTITUDE-1. So I'll probably leave extra work people need to do. One thing I want to mention, I don't know whether you saw that, John. So actually the same CAR-T has also been tested in non-Hodgkin, B-cell non-Hodgkin lymphoma. They have 100% OR, really small. Indirect evidence to show that of this one probably work not just in BCMA, but also, not just in multiple myeloma, but also in B-cell malignancy like non-Hodgkin lymphoma. And in the end, I think the real advantage based on their, the fast CAR technology is really, they can shorten the manufacturing time tremendously, right? That's number one. Number two, their cost of manufacturing should be less if they really get approved. Because they just need less cells, the material, everything, and the occupation of the facility to generate the cells is going to be less. Because when I talked to those CDMO, they told me one of the biggest costs is not really material. It's the occupation, the time they have to occupy the GMP facility. that just costs money because it's an individual drug. You just you stay there for, let's say, 21 days compared to here, they stay there less than 1 week. They just save a lot of cost. This is probably not that important in the US, but if they're talking about the Asian market, actually quite important because they are very price sensitive. That's my 2 cents. Let me know, John, and also Angus, and also Pengli. Actually, you and I cover this part. What do you think?
Perfectly, and I have nothing to add.
Okay, so actually now we're gonna go back to to fix the one we missed because I really don't know anything about it. So this is the one. John, why not you take over? the we won.
Yeah, that's right, Gonzalez. I think I had mentioned prior to the conference that we1 as a target had sort of fallen off of some folks' radar after some early results a couple of years ago from first-in-class opportunities that had seemed a little mixed. Zentalis's poster, which we caught, I think we didn't actually include it in the official review deck, more so pity, but was an interesting look at it. And it did have efficacy across a couple of interesting gyn cancers, and I would say it's positive early data. I wouldn't want to. We didn't choose it for a highlight, and we didn't spend too much time focused on it. Most of the feedback we've gotten was positive, but I don't think we didn't get a lot of incoming interest on the topic beyond the folks who were interested in the target already ahead of time. So I would say that it was generally fine data, but I would have called it a mixed response.
Okay, great. Let me quite quickly explain to you the new game we are playing. So this is our forecast, right? The prediction before the ASCO started. Now we saw all the data. We want to go back to revisit how accurate we were in terms of abstract. Oh, by the way, I just assigned the probably you disagree. The Pfizer CDK4 kind of mix. We thought it gonna be good data, but after listening to you, I thought the data was really meh. It's not that good because I learned from you. The CDK you can retreat and they still gonna have a 20% response. So now they have a 28% response after on the patient who progress on standard of care CDK. I'm not sure how good that would be, and also their tox is a little bit interesting. So I don't know. If you want to change it, we can change it back. You think you still hit it, or you think actually the data is worse than you expected?
Well, I don't think the data was worse. What struck us is that Pfizer doesn't seem super committed to any one of their next gen CDK programs or any one of the Ibrance replacement programs. And that's really what we're looking for a next step is where are they going to dedicate themselves and where are they going to start investing seriously. So I would say that the data was just fine, but it's coming in the context of, as you say, Leon, retreatment. It's coming in the context of CDK2s. There's apparently another CDK4/6 that is being brought forward in early stages. So it feels like that field is still up in the air a little bit. So for that particular molecule, yeah, I thought the results were fine. The poster itself was not bad. What we're waiting on is for Pfizer to actually make up their mind that they think they should move forward.
Okay, I will change it. Actually, so the data was good, and just Pfizer probably they look at this as good, not great data. They don't know what to do with it. But not like
exactly.
kind of
they don't know what they're going to do. We'll put it there.
Okay, got it. All right, so John, thank you very much. Actually, we somehow cover your part. If you want, I would love you to stay with us. But if you have other commitment, feel free. But I think the next one you probably are going to be very interested. Probably want to stay with us, share some comments. So we already covered this one. Oh, by the way, the game you already know what we are playing. If our prediction is kind of in line with the after data presentation, we say we hit. If we are kind of a little bit off, we mix. If we are horrible, totally predict wrong, we honestly put like a miss on it, a red flag on it. So we need to do a better job next time. Anyway, so this one already covered. I think we hit. It's good data, but we already know Carvykti gonna be a hard, kind of a bar ahead of everyone. This is good, but you just have to show us, right? Right now, idea cheap. Show me the data. I guess that's what we heard in BCMA or like a multiple myeloma in general. Even like a phase 1 good signal doesn't cut it. I don't think, out of more than 100 probably clinical stage BCMA or sort of like a multiple myeloma or even BCMA or whatever assay, if you just have some interest in phase 1, you're gonna really turn many heads. Unfortunately, just like too crowded, right? Okay, so move on to this is actually not mine. This is the Dr. Peng. You want to take over this one, please?
Sure, thanks, Liang. Yeah, so yeah, this is give us a good opportunity to clarify some confusion we might have caused last time. So there are actually 2 programs targeting EGFR and HER3. One program is a bispecific ADC, presented by Dr. Li Zhuang for a talk. And the data is I think the data are very encouraging. On the lab showing the design of the molecule bispecifics with a DAR of 8 for the ADC conjugation with the top-1 inhibitor. And I just highlight the anti-tumor response in EGFR mutation non-small cell lung cancer patients. Very good overall response rate. The safety data are also very, very encouraging. And particularly, I think this has a potential to be first in class. I might want to change to very positive if the data can hold true for larger trials. And the second abstract also presented by, was sponsored by the same company, presented by Dr. Li Chongxue for a poster. So I think the antibody portion probably is the same. The only difference, this is a naked bispecific without any ADC conjugation. I just highlight the piece of data as a monotherapy in the advanced head and neck squamous cell carcinoma as a monotherapy. The cohort is small, but I thought it's very encouraging, particularly those patients progressed on prior anti-PD-1, PD-L1 plus platinum treatment, chemo treatment. So I think it's very encouraging, again, has potential as a first-in-class drug. So the company by Li sponsored both studies. Looks like they have both assets. I think it's possible if you think about trastuzumab versus Kazila, trastuzumab ADC developed by Genentech. They both have the different applications because ADC tends to have higher, have severe safety profiles than the naked antibody. So you may have different applications, different markets. So I would say last time I was, what is the light green was a slightly positive. I probably want to change to very positive. I'm curious to hear other panelists' thoughts on this as well.
I can share a little bit. Actually, I met their CMO who are behind this 2 program. Pre-ASCO called a Pre-ASCO China Summit, sponsored by eChina Health. I actually asked exactly the same question. I said, you have 2 programs. One is a bispecific naked antibody, another pretty much have ADC. What's your plan? Are you going to advance both? His answer is yes. Probably exactly, like Dr. Peng you said. They saw this. They have so different like a therapeutic window, right? So and also the different tox profile. I would say probably therapeutic window. I don't know. Like ADC probably actually bigger because the efficacy is higher. So, but their tox, the baseline tox is just probably a lot higher compared to their monoclonal antibody. And you are right. They are trying to combine their bispecific with the other therapy like a chemo or something. That's actually give them more room to play. Because right now, in my opinion, to my knowledge, it's very few ADC can really combine with a lot of chemo, just because their toxicity overlap quite a bit. PD-1 no problem, but if you want to combine with other chemo or targeted therapy, you probably have a lot of restriction. That's what I thought. I guess that was pretty much his answer, exactly like you mentioned. That's why they keep 2 programs. But of course, they have to figure out what to move forward. And also, when I now look at data you show right here, we initially had this concern that whether their bispecific naked antibody is good enough, but it looks like definitely it's not good enough. If you look at monotherapy, they're probably even, I mean, obviously it's different indication, different treatment line, but just like to really kind of irresponsible. Cross trial comparison, you can tell the monoclonal antibody, naked antibody didn't work as well as the ADC. That's just my thought. I don't know, John. What do you think?
No, I agree that these molecules are obviously going to have different utilities. But the other comparison I think is worth making, and something that jumped out at me from the discussant in this section, is the comparison between the bispecific ADC and ADCs against either one of the targets separately. Because there are both HER3 and EGFR ADCs, obviously in this indication that people have actually looked at.
Right.
Gotten, obviously for EGFR, but even HER3 ADCs have been studied in this indication. And it seems like the bispecific really does have robustly more efficacy than the mono-target ADCs. So, in that sense, I think the strategy appears to be a success. I'll point out that HER3 as a target for a blocking antibody, so maybe for the naked antibody more than the ADC, has been a very challenging target. It's not just steric blockade of ligand binding that you need to accomplish. You also need to block constitutive dimerization, something that's not possible for every naked antibody. But with the ADC, you sort of sidestep some of those challenges with the target.
Yeah, that's a very good point. I guess for physicians, don't really care which target you have, right? If it's going after the same drug, the same indication, they just look at which one works the best, which one have the best clinical profile, right? Really, you don't have I'm not sure there's one particular subset of, let's say, lung cancer in this case. I guess it's lung cancer, right? They would have EGFR and HER3 double positive. Even you do have that, I think the size, the percentage of patients going to be pretty small. I'm not sure that how commercially attractive that's going to be if you really just focus on such a tiny part of a double positive patient. I don't know. I mean, John, what you probably know better of lung cancer space.
Yeah, remember that both of these targets can be expressed in different ways, and HER3 is expressed on a low level on a lot of non-small cell, not as a driver mutation, but as an escape mutation for others, for other therapies. So, I would expect if you're looking for a double positive, high level of overexpression, that's going to be a pretty modest patient population. But I think the efficacy of the ADC demonstrates that there's enough of this target present on tumor, even when it's not formally, IHC3+ for you to get a handle on with target therapy.
Okay, so Dr. Baum, since you changed your mind, unfortunately you're gonna get a mix because the data is better than you expected. But I think you are happy with that. Let's do that. And let's move on to next. Next, also yours, I believe. Let's just move right here. Please go ahead, this one.
Yeah, sounds good. Yeah. So this is a HER2 ADC. Leon mentioned that this asset was perceived really well by Biotech and did a huge deal with the duality. And the data is very encouraging. Particularly showed very robust anti-tumor activity across HER2 1+, 2+, and 3+. Particularly some of the patients were treated with either trastuzumab or even HER2 ADC previously. When I go through the poster, I couldn't really tell exactly what patients were heavily treated previously, but I think the data are very encouraging. So I think it's a hit.
Yeah. So anything from you, John? I know you actually talk about this. So you have some thoughts. I know you guys actually cover. I guess you cover Biotech, right?
We don't actually cover Biotech, but we obviously pay very close attention. Dr. Peng, what did you think about the safety profile? Because that was something that we were looking at pretty closely, looking for potential differentiation on ILD rates, which are very relevant in HER2 space.
Yeah, John, you asked a really good question. When I first read the abstract and then the poster, I was trying to compare the safety profiles just based on the data they provided. To me, they look very similar. The safety profile look very similar to Daiichi AstraZeneca's HER2 in HER2. What are your thoughts?
You know, I think we were maybe trying to squint a little bit to see it. Yeah, they don't have any grade 2 ILD, so maybe there's some route to differentiation there. You know, as you mentioned, there are other drugs with very similar mechanisms and similar payloads. So I think you know, unless you can find a really clear way to differentiate, good data, you know, is it enough commercial? So that's something that we're sort of thinking about.
Yeah, totally agree.
Okay. So we should move a little bit faster. We have a lot more to cover. So Dr. Peng, this one's also yours.
Yeah, so this one is super encouraging data. I know it's a small indication, rare joint tumor TGCT, but the data is so encouraging if you look at the overall response rate and the percentage of a CR/PR. So I think it's very encouraging. Of course, it's this cohort is relatively small, so hopefully the data can hold true for larger trials.
Yeah, I can provide a little bit color. Actually, I have a fortune to have a dinner with the CMO of this company. Initially, actually, I also talked to their BD to ask them, wow, your data looks like your double OR compared to the approved CSF1R from Daiichi and Plexicon. And so I really want to ask them, you know, whether there's any kind of a stone unturned. And, you know, afterwards, you know, I can tell you my understanding, the CMO is a veteran, used to work on several big pharma like Roche, AstraZeneca, GSK. So she really well respected, have a lot of experiences running, you know, kind of global trials, really kind of well trained. She's kind of a clinical developer, drug developer herself, many years. So you know, this is and also the founder of a company have a track record, already get some best-in-class drug approved on the market, small molecule. So that's the additional color I can give. You know, if there's a drug, you know, hold true, I think it would be very interesting. And I guess if you know, the big pharma look at the data, I'm pretty sure they already contact them, or they have been talking already. But who knows? Probably we're gonna see some announcement from the company soon. That's just my little bit additional color. So I guess there's another hit.
Yeah, one thing I want to add on, I think their stock went up after the abstract embargo was released. I think investors perceive the data positively, I think.
Oh, that's a really good, you know, if someone voted with their money, that's a very good validation. All right, I'm gonna move a little bit faster because we are running a little bit behind the time. So this one is really quick. Actually, you know, I am very happy I made the right call. Obviously, like I said, I evaluated this asset before. So the data is there. Actually, this the most important is the front discussion. Like I mentioned, this is a kind of a brain penetrating version of first generation of gemcitabine, right? They just tweak the chemical structure a little bit, make sure they're a bit better in terms of penetrating the brain. But all the baggage they had with the first generation EGFR inhibitor, they still had it. So that's why, you know, when they're discussing, you know, they're just pretty blunt. They said, I don't think this is going to be better osimertinib, which I kind of agree. Then they actually made our life a little bit easier. They just make a little bit comparison. As you can see here, again, this is a cross-trial comparison. It's never something, you know, we can reliably say, but people do this anyway. If you look at this, indeed, you know, you just look at this molecule, they don't compare favorably versus osimertinib, unfortunately in this case. And you know, this discussion pretty much very blunt. They said, you know, this is not something they would have considered prescribed or going to change their practice. So they pretty much say osimertinib still appears preferable. And also bear in mind, in China, there's on Chinese market, there's at least two other osimertinib, if not me better, at least me too. So the competition over there is pretty furious. So I'm not sure this molecule would have much, I would say, impact on in terms of the commercialization with this. They may get approved. I think the commercialization is going to be something remain to be seen. So I think it's a hit. And anyone have a different opinion with this one before we move on? Okay, so the last part actually going to be this part I can do real quick. So I picked the two black horses. If you agree or disagree, please let me know. This one actually, John, you cover as well. Actually, talked to the CMO of this company. She gave us a little bit more color. This is just next generation CTLA-4. The data for me, it looks pretty impressive. Why? Because as a monotherapy, they have a pretty good response in the PD-1 refractory relapse patients. I think that means a lot. If this hold true, this by itself is going to be a good market, because we know there are so many, let's say, therapies that try to target this PD-1 relapse or refractory patient, but none of them really kind of reproduce what they discover or present or what they kind of present in their phase 1 or even phase 2. Just name a few like Nectar, right? As you remember, this just didn't pan out. And this one, as a monotherapy already have this. I think there's a lot of room to improve. And obviously, they could be the same story as people had seen before. Have a good initial phase 1, I guess in this case, phase 1b data, but they could not, reproduce. But at least compared to all the others, this is a pretty decent shot, in my opinion. And also, if you look at the duration of response, some of the patients have a pretty durable response. As if you look at the spider plot in the lower left corner, and yeah, so in my opinion, I think this is something definitely worth a try. I mean, by the way, they already have their pivotal phase 3 ongoing. They also presented a trial in progress poster and this ASCO as well, and their CMO mentioned that as well. So I think this is going to be something I think worth a try with the pivotal trial. And also, the Biotech kind of in-license this one, paid the $200 million US dollar upfront and 1.5 billion total biobucks. That's kind of also validation. As we know, Ugur is a pretty good immunologist. Of course, people argue Biotech right now have a little bit kind of spray and pray strategy. Some may argue, but I think this still just looking data by itself. I think this looks one of the best molecules so far that could address the PD-1 relapse and refractory patients. What do you think, Dr. Peng and John?
Well, I would just say that one thing I haven't done yet, and really should, is put this as close apples to apples as I can against other sort of quote unquote next gen CTLA-4 approaches that are getting into late stage, like the Genesis molecule that they're obviously very proud of. Also in cold indications and in post PD-1 indications. So it does feel like there's more to CTLA-4 than ipilimumab, but I'm curious to see who ends up with the superior asset and who's able to push that forward faster. And I haven't done that side by side yet, so just something to keep an eye on. Yeah,
that's a very good point. Indeed, there's quite a bit of next generation CTLA-4 try to improve upon ipilimumab. I mean, this one at least is one of front runner. Have quite a few patient data and looks quite good. I mean, in the end, we all PhD by training. We can all find some Nature paper to tell a good story. But in the clinic, we all know the best way to cure cancer is turning humans into mice, right? But we cannot do that. So I guess the human data is the best.
To that point, Leon, I mean, Bristol's own internal next-gen CTLA-4s have seemed to stumble a little bit. They haven't seemed to go anywhere.
Right.
But there, like you say, there are several folks who have taken a swing at this, and some people have real patient data. Bristol has real patient data, although obviously not great. But a Genesis data in CRC and other places. So I do think that there are data sets out there to come, but like I said, I haven't done it yet.
Okay, So let's move another one. I'm gonna do real quick because I don't want to use too much of time. We have one bucket. So this one's another one. Actually, it's not the black horse. People or knew that, but people didn't expect the OS is gonna be so good. I guess based on there had been a little bit drag, but turned out their OS is pretty good. It's not spectacular, but for ADC, especially in this challenge indication, they read out the positive OS. That's still I would say admirable. That's definitely going to help their sales. And what I understood, their initial launch and their sales already have a pretty good momentum, and that's going to definitely make the momentum even stronger. That's for sure. That's one. As obviously. The progression-free survival have a little bit advantage. The most important eye-catching one is overall survival. They did have a respectable 0.67 hazard ratio. I think it's not the best, it's not the worst. Somewhere decent, I would say. That also means there's probably room for you to improve. And also the discussion, she sounds, I believe it's a lady. She sounds pretty, kind of enthusiastic. She's like, this is practically changing data. And presented this. I think for her the most important thing is the single arm trial, the ORR, everything translated to OS. Unfortunately, as if people remember the GSK, BCMA, ADC that didn't, right? There's a lot of also a PARP inhibitor in quite a few indications that didn't. So there's a lot of skepticism about whether the accelerated approval can really hold the water, but this one did. So I guess that's a really good encouraging sign, especially for this devastating disease. So for me, I think that's something very good. Also gave people a lot of confidence to ADC in general, right? It's not only just reduction of disease progression, but also the overall survival, which is the gold standard. So anything you want to add, Dr. Peng and John, for this one before we move on?
No, I think you hit the nail on it.
Yeah, nothing from me.
Okay, so now move to the last bucket. This part, Angus are not gonna fake doctor again. Unfortunately, Dr. Chandler Park, because of some emergency, couldn't join us. But we spent a couple hours learning up all those abstracts, so I think we are good. Angus, you want to take it the first one? I know I actually wrote quite a bit about it, so you're actually an expert.
Right.
Please go ahead.
Right. So this is the Indigo study from Serbia vorasidenib as potentially from the different researchers and investigators I've heard, they said this might be the first targeted therapy for low-grade glioma. So just remind everybody, this is an IDH1/2 inhibitor, Serbia bought it with Gio's oncology portfolio. This was, I think, 2 to 3 years ago. The portfolio also involved to approve the therapies, including an IDH1 inhibitor and an IDH2 inhibitor, which is partnered with Bristol-Myers Squibb. So about this study, it compared vorasidenib versus placebo. I think grade 2 glioma with IDH1/2 mutation. These patients had no previous treatment except for surgery. And for those who wonder how much of the population is IDH1/2 mutation represent in the overall population, is about 80%. So I think the data are generally very impressive. The PFS reduction reached 61%. I think everything above 50% can be safely say is very impressive. The median PFS, as you can see, more than double the median PFS for this population. And researchers, I think the commenter, ASCO invited to a commenter said that they all said this is kind of practice changing during our press briefing. Actually, they said this practice changing clinically meaningful. It could lead to this approval. There is one slight small concern. You could make a big thing out of it, but there is a liver toxicity with ALT elevation of 10% of grade 3 or above, and there are also 2 relatively serious cases where you could think of it as a signal for potential liver failure. But both Serbia and the investigator said these events are reversible and manageable. And there is a fast track designation from the FDA on this indication, and I think Servier is targeting potential approval in 2024. They haven't filed it yet, but we can look at the approval by 2024. That's it on this one.
Okay, so there's a clear hit. Let me just
Yeah,
fix
I think
that
this
first
is
for you.
There were some concerns reached about the talked about the liver toxicity, but I think generally speaking, people aren't so scared about them.
Yeah, so the next one actually is mine. This one, there's no new drug involved. It's just like a really old school chemotherapy, radiotherapy. They just like shuffle them, try to say which one work better. Simply speaking, this for this particular kind of early stage rectal cancer, the previous standard of care involve a pretty heavy dosing of radiation therapy, especially when you do radiation around your pelvis area when you are young. It's not very good. Several downsides, right? If you are young, your reproduction organ going to be destroyed. So you lose your, childbearing capability. Also, you destroy your bone marrow. So you have a much higher chance to get a bone fracture. And pretty much is a lot of stuff. And also you probably get a secondary cancer because the radiation, as you know, could have that. So the idea for this one is, can you replace the standard of care and the time radiation with a kind of intense FOLFOX 6 cycle of chemotherapy by itself? If for those a good responder, right? So they could just proceed with the surgery. That means like you have a more than 20% respond, I guess, by the shrink of the tumor. If you did not respond very well, you go back to the old one. Just have like an older, chemoradiation. So pretty much, the idea is, really right now after they understand the chemoradiation and also chemotherapy better, probably less is more. That's the whole idea. So the idea they designed the trial is like a non-inferiority in terms of efficacy, and try to see whether they can improve the quality of life and also reduce the toxicity. And they did prove that. First of all, if you look at the overall survival, almost no difference. This is really just like kind of identical. That's number one. Number two, I mean, there's a lot of data, right? I just picked the two most important one. If you look at quality of life evaluation, you did say a big difference. I think the most important, if you look at like I mentioned, the rectal area, if you do radiation, can destroy your reproduction organ. If you look at the chart in the lower lower left, if you look at the lower two panels, look at the male sexual function and the female sexual function. There's a lot of improvement compared to the old one, right? So that's a very good. So that definitely, that's why in the discussion part, first of all, this paper was published in New England Journal of Medicine, and also the discussion, well, actually, she's a radiation oncologist. They invited her and her to say that, those patients should not receive radiation oncology, and she agreed. That's something she supports. This is a really good summary of, this kind of a fine balance, right, between toxicity and efficacy. So it turned out the efficacy are very comparable, but toxicity of this one is definitely favorable versus the old standard of care. So this is definitely something like a practically changing. But of course, it's not every single one of them. So the breakdown, still probably minority patient can still receive the traditional chemo radiotherapy. For example, some patients just could not tolerate a 6th cycle of FOLFOX, then they cannot be taking this one. So this one is a kind of a clear hit, and so I can move on to the next one real quick. So what's next? This mine or? Oh yeah, this one also actually a clear hit. A little bit history about this one. Actually, this relates to AstraZeneca, and also there's a lot of chatter about how AstraZeneca is heavy on China. There's actually a really good story about this. So ADAURA phase 3 trial has been going for a long time, and it's a really good collaboration. There are 3 lead PIs, one from Japan, one from US, which is from Yale, and the one from China, which is the #1 lung cancer doctor in China. So they did this a really good trial. And, they already published this in 2020, the primary funding. Actually, the first author is Yilong Wu, who is, by the way, the biggest lung cancer doctor in China. So that data was really good. And, the primary measurement at the time is the disease-free survival. I mean, there's clear hit of that one. That's why it get approved. But still, as you probably heard for targeted therapy, the biggest challenge is does progression-free survival benefit translate to OS benefit? I mean, there's always some critics said, okay, you can only delay the disease progression, but you will not have a much improvement in terms of overall survival. And this data really show that you did have a benefit of overall survival, and the hazard ratio looks fantastic. That's all good news, right? And from like a trial design, results, everything, and also from a commercial point of view, because a lot of patients from this trial were recruited in China, so this would definitely help the penetration of this drug, or probably help get this drug be covered by a lot of insurance in China due to this data, right? That's very good. There's just one tiny, kind of a diverging voice from this regarding the trials from this guy called Vinay Prasad. He's the long-term critic of Big Pharma. He just don't like Big Pharma, let's put it this way. Just I want to give a little bit background about his position. He's a very, very vocal critic. He challenged the ethicality of this trial. His argument is like, well, AstraZeneca, you probably already knew this is going to be benefiting the patients. Why you didn't give the patient the control arm this drug and to let them kind of lose many years? So,
I have to say something about this. I mean, I've heard the investigator. I forgot his name. Anyway, I think that 90% of patients eventually got some kind of EGFR inhibitor in the placebo arm after they progressed. And in those, I think that if I got the number right, about, I forgot it, I have to, don't quote me on this, either 20% or half of them actually got Tagrisso in the placebo arm after progression. The point is of this, definitely there is no OS benefit, but there is definitely an ask and a request to see the data among direct comparison between the osimertinib versus those who got Tagrisso in first-line advanced disease setting. That direct comparison, you don't count people who got other EGFR or those who didn't get EGFR. Some people still wanted to see that direct comparison to really understand whether you need to give Tagrisso early on in the adjuvant setting, or whether it is okay to just wait until you may get the same benefit in the metastatic setting. So direct comparison, osimertinib adjuvant therapy versus people in the placebo arm who didn't get it in the adjuvant setting, but got it in the advanced setting. We don't have that subgroup data available yet. Yes, Dr. Herbst. Yeah, I remember that. Roy Herbst. So he was just saying they're following people longer just because Tagrisso wasn't available. This trial lasted a long time. Just some people weren't available. Tagrisso weren't available to them. But for the rest of patients who have yet to recur, those people will likely, I think he said 400 patients, again, don't quote me on this, who didn't still have yet to recur, and they would expect these patients will get Tagrisso in the first line metastatic setting. I mean, he said that these patients are still being followed. They will probably have those data, direct comparison Tagrisso adjuvant versus Tagrisso metastatic later. That's it for me.
Yeah, so you are pretty much you kind of disagree what this guy's
I do, but I do acknowledge that they didn't completely answer the question, because we still need to see, like I said, the direct comparison group. I mean, this OS data is a mixed bag, meaning there are patients who didn't even get a EGFR, about 10% of patients who really didn't get any EGFR inhibitor afterward. And also we know Tagrisso just worked best among the EGFR class if you are giving some of the patients Irisa and other EGFR inhibitors. We don't know how that impacted patient survival. So there is definitely a need for us to see the direct OS comparison between Tagrisso adjuvant versus Tagrisso metastatic. So they don't have that data yet. Yep.
I see. Yeah, first of all,
but he's claimed that they didn't give them the drug. That's a little bit
You know, some people just disagree or criticize him press, right? I mean, I follow him a little bit. No bad blood whatsoever, but he has a huge number of followers on Twitter. Fortunately or unfortunately, right? So this is freedom of speech, I guess. You just have to be sophisticated by listening to some. Not every MD from like a prestigious medical school gonna have the most comprehensive knowledge, in my opinion. So that's a good example. Then there's a lot of chatter. You probably heard about that. Let's just move on. Next one. In the meantime, we just put this one as a hit. Definitely a hit, no doubt, despite the little noise. This one, I guess that was for Angus.
Right. This is a collaborative group, National Cancer Institute sponsored study. I think there was a data leak on Twitter. I didn't see it, but I heard people talking about it, about BMS PD-1 inhibitor versus Seagen's antibody drug conjugate, etc. in first-line classical Hodgkin lymphoma. Obviously, there was also a big progression-free survival benefit of Opdivo versus Adcetris, this reduction of over 50%, once again, a large PFS benefit. And also on the safety side, there was also a big benefit for the PD-1 inhibitor. Even though neutropenia was sort of higher, the rate was higher with Opdivo versus Adcetris, there's still fewer people on the Opdivo arm who received the GCSF for the neutropenia versus almost all patients got it in the Adcetris arm. There were also fewer pain cases. And I think the investigators highlighted the sensory neuropathy thing as very meaningful to patients. Opdivo had lower rates, and also the treatment discontinuation rates for either Nivo or the ADC. The Nivo arm also appeared better in terms of patient discontinuation. OS data was immature at this time. After just 1 year of follow-up, you really couldn't tell anything at this point, even though it still slightly favored the Opdivo arm. Just a reminder that for Adcetris, it recently had NCCN Category 1 recommendation because of a OS benefit shown in this Echelon-1 data. And that OS signal came out after a median 6 year of follow-up. So I think this is going to be a long time until we see that. I had a chance to interview Lady Stain, Seagen CEO at ASCO, and he was like, we want to see the OS data. Of course, we want to see the OS data. But that's what he wanted to see to really see the benefit. But the doctors I've heard, they were like, this PFS benefit and this safety tolerability benefit is already good enough for them to start switching to Opdivo in first-line chronic Hodgkin lymphoma. But there will be some patients, I think, I read someone saying that some people with underlying immunological disorders, doctors might keep them on Adcetris versus Opdivo, which moderates the immune system. And also a reminder that Adcetris is now Seagen's top-selling drug, but still that will soon be replaced by Padcev. And Seagen is also testing the combination, which I didn't hear a lot for some reason, people discussing this, the combination of Adcetris plus Opdivo in this setting. John, if you have anything to add about this?
Yeah, I think you did that was a fabulous recap of the trial. The couple of quick things that I would just add on top of that is that, of course, Seagen is testing chemo plus Adcetris plus Opdivo. They're running that phase 3, but the control arm in that trial is, of course, Adcetris chemo, not Opdivo chemo.
Opdivo chemo, right.
So now the control arm is now the wrong control arm. So obviously now they'll have to be doing some cross-trial comparisons to convince people to get back to giving Adcetris in the front line. So that's, I think that's an interesting point. The one other thing that I would mention, well, and I think Angus, you said this very, very clearly, the docs are absolutely convinced, right? A plenary at ASCO, very strong response from everybody in the room. I haven't spoken to anybody not working for Seagen or heavily invested in Pfizer that thinks that this isn't practice changing. So really great. And Yeah, maybe the one other point to moving beyond just the practice-changing element of it, and maybe more on the investment side of it, is of course that with Pfizer proposing to buy Seagen, the value of this to Seagen doesn't really matter. Pfizer's very committed to the deal; they'll pay the price. Seagen is where it is. But what people think of that deal from Pfizer perspective is very much on the button here. And this was only ever expected to be 1 to 1.5 billion revenue by 2030, out of 10 or 11 that Pfizer was talking about getting at Seagen. So not the biggest driver by far of the deal price, but it does call into question the fundamental assumption that Seagen ADCs have effectively infinite lifespan, right? That was not something that people said very explicitly, but it's sort of implied in the deal price and the Seagen valuation, both that people were talking about for Merck deal and Pfizer deal, that you're not going to be able to erode these drugs very easily. Most people are thinking about the IP side of things, the manufacturing expertise side of things. But it's worth saying that folks don't necessarily account for the fact that the fields themselves move on, and the investigators are going to run these trials and find new standards of care. So you can't necessarily give 20 years worth of credit to any drug. So, but this was a very big splash at times.
I was going to say, just it's interesting to see how these two big ADC complete M&As, acquisitions, Gilead, Immunomatics, and Pfizer Seagen didn't exactly. I don't think they're going to get their money back, whereas you have the AstraZeneca direct licensing deal seems to be working really well. This is an interesting dynamics to see here.
It's a very interesting dynamic. I think, even as recently as a year ago or 2 years ago, the assumption was that ADCs were very difficult to make, and Seagen had some sort of secret sauce, and they were going to be able to put these things together in a way that nobody else could. But even just at this ASCO, I think it's clear Seagen's very good at making ADCs, but they're not the only ones that have that ability.
Right. I think that another point I want to raise, I think from my interview with David, was like he said, of course, there are those candidates that you could see from their pipeline. They have a lot of things working behind closed doors that we just don't see. And also the combination of Pfizer's chemistry capabilities with Seagen's biologics capabilities, they think they could potentially get some of the better ADCs in the future, ADC platforms in the future.
No, I think that's very reasonable. And not to knock either company or their expertise. They're heavyweights and they're going to make great drugs. But this is sort
right
of
now. This is definitely negative.
This is not great news. This is not good news, even if you believe in the platform.
Yeah, if you put this into the context, if you look at totality of all the ADC clinical data and ASCO and AACR, you can see clearly the innovation center is not in the West. I mean, that's just, I could be biased. You just have to look at data, right? The phase 1 data, those are leading molecule. Most of them come from Daiichi, from like some of those China biotech, kind of like a duality, right? Like, this Bai Li, right? They have pretty interesting stuff. One thing, I actually did a research. If you look at the basic research in chemistry, East Asian countries, 3 of them are leading in the world by a large margin. I mean, ADC is really just a chemistry, medicinal chemist, right? That's, the biology target, yes, is important, but once you have it, the best in class or fast follow is like whoever have the best chemist, right? Figure out the linker, payload, those kind of stuff, actually kind of better advantage. Unfortunately, for a lot of, even for Seagen, right? They don't have the most advanced platform. I mean, John, you and I probably did deep dive on those kind of ADC platform, right? Daiichi probably have established one of the best platform, followed by a bunch of other Asian biotech, like even like South Korea have a LegoChem. They probably have a pretty good, platform. So, let's say, but Pfizer is definitely much, much stronger in terms of commercialization. As we know, we learn from PARP inhibitor space, if a very strong commercial team, you may still hold up. I mean, I think David was partially right? Show me OS data. They can still use their marketing team and try to persuade a lot of community oncologists, right? The teaching hospital definitely gonna switch, but a community oncologist show me OS data. Often that indeed was the case. who knows? Let's just finish up. I know we're running over our time. It's like, hey, this one is easy. It's the Zuma-7 data already approved many years ago. Now just coming the OS data, right? It was approved based on the PFS, event-free survival. Many years ago, that's New England Journal, not really many years, actually last year, New England Journal Medicine paper, right? That looks pretty impressive. That's another, really great kind of data to show that there's a newer generation of molecule. They have an initial progression-free survival, event-free survival benefit that translated successful to OS, which is very encouraging, right? So this is another great example. But of course, the hazard ratio drop. I mean, the later line, they probably converged for those patients. They're very likely to receive the same later line. So they converge, that's expected. And also, I think the most important take-home message is like right now, it sounds like those physicians really kind of very willing to accept those CAR-T move to front line. Actually, in this discussion, I think this one is very interesting. It's not necessarily for like, the CD19 Keytruda, but they're talking about a Carvykti. They show this clear, improvement of benefits when Carvykti move to early line. I think that the medical community is accepting that. They're already ready, just like what they did with the PD-1, right? Start with the last line, and they get into the front line. They even get the PD-1 into early diseases, right? Stage 2, stage 3, right? So I guess it looks like CAR-T are following the same trajectory. But of course, the bottleneck gonna be, like we mentioned already, the CMC, manufacturing, the logistics gonna be always there. But I guess those problems gonna become smaller and smaller over time, hopefully, because the benefits are very clear, right? You move to earlier line, then somehow the patient benefit better. What do you think, John? What have you heard from those from the investor and also from the company?
I think what's holding back CAR-T is generally two things. One, it's the harshness of conditioning regimens, and two, it's the expense and challenge of manufacturing. And that was always the feedback we got from docs, is that, yeah, the benefit will be better as you get to early lines like anything else, but inevitably there will be a balancing act where in very early lines, especially for larger indications, it's easier and quicker to give antibodies or to give small molecule combinations. So taking, for instance, multiple myeloma, right, where we've got 5, 6 lines of therapy, where you routinely see patients in clinical trials that are 8 or 9 prior lines. At what point is Carvykti going to displace Dara, Rev-Dex in front line, right? It's just so easy to give those triplets and quadruplets, and it's inevitably going to be a little bit more challenging to give CAR-T even once it's fully mature. That the expectation is that probably you can't move too much earlier than CARTITUDE-4. Second line, third line, that's probably as far as it goes, or at least that's what the expectation is at this point, I think. We'll see. For indications where there is the potential for a curative or semi-curative CAR-T, where you get really, really long durations of response in what would otherwise be aggressive indications, where you're almost replacing transplant, maybe. There, possibly you could end up in a first line and a frontline setting, but probably with bridging therapy, probably with aggressive conditioning that will limit the population size to those patients that can tolerate it. So there'll be a balance, I think.
Yeah, I want to throw another kind of a ratchet into the mix. So if you look at the pricing of, let's just compare Carvykti and TIGIT. Carvykti is over 400,000, I believe, right? So the TIGIT, the 1-year treatment cost is almost 400,000 already. If you think about it, and also like a Carvykti manufacturing cost is a lot higher, so their profit margin lower, right? Let's say per the 1-year treatment of TIGIT, the profit J&J gonna make compared to 1 dose of Carvykti, I think is comparable, more or less, if you remove the COGS and everything, right? not to even mention the capex of investment of all those manufacturing facilities. You put that in there, the TIGIT is a clear win, right? The dose is so low. The dose they give to the patient, the total amount of material is much lower than PD-1. Right. So if you consider this, let's say, I always use this, you can correct me, it's a really kind of a last comments or discussion. If I have a patient who are like long-term responder to TIGIT, let's say for a couple of years, from financial point of view, right, J&J actually make more profit out of that compared to CAR-T. I don't know whether they're going to consider this. Obviously there should be like a doctor decide on this. But I think if I don't know whether commercial team gonna have a liberal preference. I don't know. What do you think? Did actually investor ask this question? Because I mean, for me, it looks like there could be some math over there.
I mean, it's a pretty cynical question.
I think I asked last year at ASH their kind of strategy of whether doctors I've heard last year at ASH were very disappointed in how J&J performed just because it was already number 4 into this game. They expected, they know the ropes, they should be doing well. So I asked them, why aren't you doing this? Has TECVILI sort of distracted you from devoting 100% into CAR-T? And they were like, no, this definitely, they have separate teams. Both of them are working 100% on each of those therapies.
I would expect the teams to end up being separate. And insofar as J&J corporate might have an opinion on this, at the end of the day, J&J wants to own first line and second line and third line and just pass patients from therapy to therapy. I think the difference in margin is probably there. It might even be meaningful, but it's much easier to get docs on board if the data supports the sequence that you're going to suggest. It's much easier to take and hold market share if the docs.
Yeah, looks like J&J is really on the track of a winner take it all, right? Every single line they have something. I think right now the BMS only have a Revlimid, which is going to be off patent soon.
Already genericized, right? They had volume gated entries starting last year. Yeah. So let's say going to be very interesting. All right. So thank you guys for so much for staying 20 minutes over time. So really appreciate John and also Angus and Dr. Peng. We can take one question if anyone wants us to ask a question. If you type in the Q&A or raise your hand, that probably is only one question we can take. I'm gonna give you like 30 seconds. In the meantime, while others are waiting for this, please follow us. Right, this is the we are too tiny nonprofit. In Science We Trust community is such a cheesy name. We are PhD actually right now. Angus, I think you can get honorary PhD because your comments are oftentimes better than a lot of PhD. No, but what I said just a joke, right? I really cannot give people a degree. But please follow us if you want to follow us on LinkedIn. If you want to join us on mailing list, that's on the left side, the Science We Trust community. BioSpark is more established, have been around for more than 7 years. So we're more established. If you want to join our WeChat group, scan the BioSpark admin, she will help you. If you want to visit our website, look at the replay of, I think right now probably over 100 webinar we have done. John, if you are interested, we can give you a free membership, by the way. You can have a little bit time because you contribute. That's how we do help, kind of show our courtesy to our speakers. So again, if there's any, there's one question. Let me say what the question is. What did you miss? That's a quick, kind of interesting question. Anyone want to take a swing? Well, I don't even know what that means. I mean, there are definitely things that I missed. There were, it's an enormous conference, and Omer and I were both on the ground there, we had a team of 4 people in the New York office, all looking at presentations, saving down posters. There's a ton of stuff I missed that I only found out about listening to my colleagues on our own ASCO review webinar. So there's just way too much for anybody to take in, really, at the conference, no matter how dedicated you are. So I learned all sorts of stuff about the TROP2 ADC space that I had not been paying attention to at the conference because I just was doing other things. I saw a bunch of things elsewhere in the CDK4/6 space that I hadn't caught at the conference, that I only found out about after the get back. So there's there's a lot there, and too much for anybody to get.
I think
how about each one of us? Go ahead, Angus.
I have to say I wasn't able to go to even one session just because I had so many interviews with everybody, back-to-back interviews. I didn't even have time for any of the sessions. But I just wanted to say one kind of a high-level observation based on our press briefing, at least, is that there is a general sentiment around doctors to find the right patient for the right kind of therapy, meaning they want to de-escalate, as the one that you talked about, Leon, that I think the prospect data, they want to de-escalate therapy for some patients while identify those patients who need some stronger regimens. So they want a more precision medicine approach to things. That's something that I took away from this meeting. And the second point is, there is definitely a need, as John mentioned, in that Adcetris plus Opdivo versus Adcetris instead of Opdivo study, that there should be some kind of industry should start thinking about how do you ensure that your study design is relevant when it actually reads out like 3 to 5 years ago. Just because patients are living longer these days while our industry is moving so fast, the trial design you have today might not even be relevant 3 to 5 years from now when you read it out. So we've seen this with AstraZeneca with their Imfinzi Lynparza trial. I mean, the trial investigator she said during our press briefing that she wished they had an Lynparza therapy arm to kind of tease out Imfinzi's contribution to that dual regimen. So you started to see more of that lately, where they wish they had another trial design when they actually read out. So this is, I think, also a trend that's going on from what I'm seeing in that space.
Dr. Pong, what's your closing mark? What did you miss?
Well, I echo what John said. There's such a wealth of knowledge from the conference. It's hard to cover everything. This is the highlights. It's not try to cover everything also.
Yeah, definitely echoes. So everything. I also have to admit, I was busy with the meeting people. I didn't go to any session either, but I did follow them remotely. I have to say, especially after the ASCO, I watched a lot of presentation myself. So my ASCO actually lasted until today, especially when I prepared a slide, I listened to them. There's a lot of information. I think actually, if you have access to them, re-listen them. You will find that there's so much information missed when you first listen to them, right? So, people spend a lot of time prepare for the presentation. There's a lot of information you probably missed. So I highly recommend it. Recommend you to revisit those key presentations. You learn a lot more. Anyway, with that being said, thank you very much. Thank you especially the speaker, Dr. Pong, and Dr. Mailer, actually a doctor, and Angus, Mr. Liu, Mr. Angus Liu. And also thank you, the organizer from BioSpark and SAPA GP, and our very, very dedicated 5 median promoters. Thank you all, the audience, spend the time with us on Friday night or Saturday morning. Not show your dedication. Hopefully, this time is well spent. I look forward to meeting with you again next time. Thank you very much, and bye for now.